From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on February 12th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in chilly Tacoma, Washington. Today is the 79th episode of Matters Microbial. Thanks as always for listening, viewing, and sharing the good microbial word. On to 100 episodes? I hope so, and I'm grateful for your support. When I first started this podcast, I would sometimes share strange and wonderful things I have in my Wunderkammer, my cabinet of curiosities. I try to keep that to a minimum, except when it's microbial. I'd like you to look for a moment at this image of a stromatolite of the Maramamba tiger iron formation from Western Australia. The different colorful layers are the results of layers of ancient cyanobacteria as the oxygen levels dipped and swelled so long ago, precipitating iron as they went. It was a gift sent to me by a friend who wanted to brighten my mood. That's right, fossilized bacteria 2.7 billion years old. And that certainly gives me perspective in these strange and uncertain times in which we live. I will put links to this formation and stromatolites in the show notes. I've collected fossils for many years, and here's a typical fossil of a trilobite. These lovely arthropods ruled the ocean 500 million years ago. But what did they really look like? In some environments, low in oxygen and high in sulfide, the soft parts of these invertebrates can be replaced by fool's gold, iron pyrite. This specimen of a trilobite over an inch long is from the Beecher Quarry in upstate New York. Close up, as now, you can see legs, gills, and other soft parts of the trilobite, including eggs. I will include a link to the Beecher Quarry shales. Now, you knew there had to be a microbial connection, and yes, there is. There's some evidence that trilobites formed a symbiosis using autotrophic sulfur-oxidizing bacteria, as shown in this paper. And this is based on the structure of legs and other soft body parts, including feeding parts, that are seldom preserved during fossilization. I will include a link to this fascinating paper. Today's podcast topic is an unusual one for me. After all, I've chided educators on this podcast for their eukaryocentrism many times. Eukaryotes, organisms with nuclei, like you and me, versus things like bacteria and archaea. And some of the first buttons I ever made, seen here, promoted prokaryotic pride. Now, that expression is somewhat controversial in the microbial world, since it's felt it might exclude archaea, but I think all of you who know me better than that. Why not look at single-celled eukaryotes? And most of us have in one form or another. Amoebae, paramecia, even the scary ones like the brain-eating amoeba Naegleria. They are fascinating and relatively simple to visualize, even for old eyes. And some of these single-cell eukaryotes, these protists, are extremophilic. Incidentally, today's guest has a coworker who creates glass artwork based on their interests in the microbial world. Since we're celebrating small eukaryotes that are extremophilic, why not appreciate this glow-in-the-dark glass bead tardigrade a friend made for me? Most work with extremophilic microbes have focused on bacteria or archaea, but what can we learn from these extremophilic eukaryotes? So, it is a genuine pleasure to introduce Dr. Angela Oliverio to the podcast. Dr. Oliverio has many interests, and today we'll discuss extremophilic single-cell eukaryotes and what we can learn from them. Welcome to the Quality Quorum for today, Angela. It's wonderful to meet you.
Welcome, Mark. It's great to be here.
I am so excited, and I have to tell listeners and viewers, this is definitely, when you go to the show notes, you have to look at the website, because there's so much cool stuff going on every single level.
Oh, I appreciate it. I always feel guilty, like I should update it slightly more often. So I Love that feedback.
Well, I mean, I won't even talk about my own. But what I would say to you is that maybe a good place to start is why eukaryotes?
Yeah, absolutely. And it's so funny, Mark, because as you were talking about the sort of groaning at the bias of eukaryotic study that many microbiologists feel, I feel that too. And I'll say that microbial eukaryotic biologists, which are also called protistologists, also feel that way, perhaps even the most of anyone, because microbial eukaryotes are in this sort of funny silo, right? We're eukaryotic people, study plants, animals, and fungi. By the way, the fungal biologists have done a really good job of convincing people that fungi matter. So I always like to say, oh, protistologists need to learn from them, right? But we've done a really good job learning about eukaryotes, and we've done a good job learning about prokaryotes in the past couple of decades, right? And I think that there's this funny gray space in between, which is the microbes that fall in this other category, right?
What's interesting to me is that in elementary school, even, most places that use those really cheap microscopes that we all have access to, and we should have better access for young people. But when they look through that microscope at a drop of pond water, what they're generally looking at are the protists, these single-celled eukaryotes. And it's interesting you used the word protist. Go ahead and say it one more time because Lynn Margulis is in my head.
Yes, protists.
Protist is the name, and people who study it are.
We like to avoid the word protozoa because that's
Yes.
like
An animal, right? Exactly. And so for me, it was like euglena, amoebae, paramecia, my very favorite blepharisma.
yeah.
All of these things are beautiful to look at and fascinating to see. And I'll put some links in the show notes because especially for people who were I call them new micronauts or novice micronauts, so many cool images just from that, even though your work is with ones like that live in unusual environments.
Yeah, that's right. And it is this funny tension, right? Microbial eukaryotes are some of the easiest microbes to see and visualize, and a lot of the work over the past centuries really started there. But then we've started lagging behind in the update with sequencing and omics, and so now there's this gap, and that's kind of where our interests lie in filling in that gap and bringing what we know about microbial eukaryotic biology into the same world.
I mean, for me, it was different because when I was in college and in graduate school, I didn't fit in very well. There's a surprise. And so some of the emeritus professors who still did work with protists were still around. And I would just sit and listen to them because they could see things that I couldn't see. I mean, when you normally look at a bacterium or an archaean, you don't get to see the internal structures without very specialized equipment, as we talked about last week on the podcast with electrocryotomography. But in many cases with protists, you can actually see some of these wonderful organelles that serve the function of different organs in us.
Yeah, absolutely. I totally agree. And this image of protist diversity. So this is a panel showing kind of just one snapshot of one organism from each of the major protist supergroups across the eukaryotic tree of life. The morphological diversity is stunning, right? And I think that's one of the first things that we really appreciated about protists. And now we're realizing how much more we can learn about eukaryotic biology writ large by studying them, right? So one of my favorite examples is there's this ciliate that, was discovered to have a different genetic code. That's challenging sort of a fundamental rule of biology, right? And then it was discovered in one ciliate lineage, then it was discovered in another ciliate lineage, and then another. And then people were like, oh, ciliates are weird. And that's kind of where we left it. And then recently, another genetic code was discovered in this group called Heterolobosia, which is another large group of, eukaryotes that no one's ever heard of, right? And they're not related to ciliates. I mean, they are, but they're very distantly related to ciliates.
Sure.
So now we're seeing that these sort of fundamental rules of how we understand biology are being challenged. And it's not just one weird group, it's across the tree. So it really matters what types of organisms we study, our understanding of biology. And so I
So
think that's kind of like some of the exciting genomic stuff that we see.
I see it in the news a lot. I don't know the name of the protist. This is the one with the long neck. You know, the one that I'm talking about, that like a snake strikes at prey organisms. I will put a link to that. That video is amazing. But the point I'm trying to get at is there's so much diversity and so many interesting. You mentioned, of course, the slightly different genetic code in Tetrahymena and how that in turn led to a Nobel Prize, which is always nice to think of.
Yes, right? Big ambitions here. Let's go, right?
Right.
And I wanted to show another image sort of on the topic of protist diversity. So this one is an image of a testate amoeba, or a shelled amoeba, which is one of the first protists that I ever studied. And it's incredible, right? These single-celled organisms are producing these intricate shells. And so here you can see this is a difflusia, which is a particular lineage of testate amoeba, and it's actually a big predator of the microbial world. And it doesn't just feed on bacteria, which is what we often think of protists as doing. It's actually feeding on diatoms. And after it feeds on diatoms, it's secreting cement to make a shell around its cell, and then sticking diatom frustules to build its sort of armor, right?
So what you're saying is they're putting the skulls of their prey onto their
That's
outer
right.
surface.
That's
That's
right.
that's very metal.
I'm going to start saying that.
Please do. Please do. And you know, it's interesting too, because so many diatoms have those glass frustrals. And I've
Mhm.
often wondered, so do those just get exported to part of the armor on the outside? They're not being
Yeah.
internalized, are they?
As far as we know, no, but yeah, and it's so funny, right? Like for a long time, folks thought that the shells served as a defense mechanism that
Yeah.
amoeba could hide in. Uh, Recent evidence suggests that they're actually used for predation. Um, and there are some really cool ones. I don't picture this, but that they have spikes on the
Yep.
aperture or the shell opening, and that's actually like how they're hunting their prey. So I think it's
So
kind of
how is this not punk rock metal?
I
Come on.
know. Exactly. I try to say protists are like the lions and tigers and bears, at least
Yeah.
the ones that consume prey of the
We need to
microbial
come
world.
back with nature being red in tooth and claw. We need to change this to pseudopods and maybe frustrals or something similar like that, because that's what we're dealing with. And this is important for listeners and readers. Yes, I get excited and enthusiastic about things. I do. Guilty as charged. But the three most important words in science are I don't know. And that's an opportunity, not a criticism. And often we find things that we never expected would
Mhm.
have real relevance to everyday life. And it's really important for listeners and viewers to understand this. It's very common. My late parents would say, why was I studying root nodule bacteria? And I had an answer for that. It, having to do with nitrogen fixation, though I didn't work on that. But at least they understood that. And I wish we lived in a world where people could understand the joy of investigation in and of itself.
Absolutely. I totally agree. And I think that there are so many good examples of weird random investigations leading to sort of the next big discovery, right? Whether it's Thomas Brock studying extremophiles in Yellowstone and discovering Thermus aquaticus and sort of the foundations of PCR and sequencing technology that's now ubiquitous across biotech and science writ large. right?
And that was, again, remember, help discovering that with Tom Brock was his undergraduate student with the best name in all of science, Hudson Freeze. That's a
Yes,
supervillain name.
that's so
And
good.
he's actually a cancer biologist in San Diego now. But can you imagine being the undergrad who actually helped find the organism that was the center of PCR later on?
No.
So this is what I want the public to see.
Yeah, I agree.
So I'm too excited. So let's get back to what you're looking at.
Yes. So hopefully, I've convinced you that microbial eukaryotes are a good place to start in terms of major sort of gaps in biodiscovery, right? And I
Yes.
think then the question is, okay, well, where should we study microbial eukaryotes? And I think that extreme environments are an obvious first choice. And I say this because if you think about where so much of the novelty, microbial novelty has come from on the prokaryotic side of bacteria and archaea, it's extreme environments. I think
Right.
almost all of the candidate phyla radiation was discovered from extreme environments, much in geothermal springs. And this is not by accident, right? By design, these sort of extreme physiochemical conditions push evolution to its edges, and sort of the blueprints of metabolisms and all of this sort of crazy biology we get to see in extreme environments. So while I think that protists should be studied across all environments, our lab targeted geothermal springs as a place to start.
So I want people to understand when we use the term extremophilic, we're using it at least partly today in terms of higher temperatures, and that has all kinds of effects. And the organisms in turn, it's like the red queen of evolutionary biology with adaptations that teach us about things. Are there other forms of extremophilic conditions that you're studying as well besides temperature?
So we're really focused right now on temperature, and that's
Okay.
partly because we discovered, and I'll tell you about this in a little bit, a really cool organism that can survive hot temperatures. We're also really interested in pH, low pH, so super acidic conditions, and the intersection of those two. So we
Yes.
call that like thermoacidophilic environments, because things get crazy when you have to adapt to multiple extremes, right? Um, Certainly, many other types of extreme environments, extreme radiation, extreme cold, extreme alkalinity, all really fascinating areas of research. So we're just focused on one sliver. And I can say a little bit about why we focused on temperature and why we focused on amoeba
Sure.
in specific. So as you can see from this figure that's looking at temperature and pH optima across different eukaryotic lineages. This was a paper that came out of our lab a couple years ago. When I was first starting my lab group at Syracuse, we didn't have a wet lab yet, right? And I had a tech at the time. Now she's a PhD student, Beryl Rappaport, who is actually the one that does the glass work. So I had her start on the same day as me, and then I was like, oh my gosh, what did I do? There's no lab for you to work in yet. But it was a blessing in disguise, and she started tackling this project, which is, well, what do we already know about eukaryotes in extreme environments, and specifically focusing on microbial eukaryotes or protists, all of these lineages that make up the vast majority of phylogenetic diversity across the eukaryotic tree, but are quite understudied as we've discussed, right? So she pulled together all of this data from different publications, kind of documenting, which surprisingly no one had actually done, the temperature limits at the high end and at the low end, and same thing for pH and salinity. And one of the really interesting things that she found is although there's a fungus and a red algae that can survive 60 degrees, which are fairly well characterized, surprisingly, there were all these amoeba interspersed at high temperatures. And each
I
amoeba had only had like one paper from, a decade or decades ago, kind of describing it, but for which no genome existed and for which we couldn't get the culture anymore. So we're like, okay, we know that these amoeba exist. We know that they're really good at high temperature. We have no idea why, and no one does. So that was kind of the question that we were left with at the end of this exercise of compiling information on extremophilics.
I wouldn't have guessed it'd be the amoebae, because when you look at things like paramecia or tetrahymena or blepharisma, or may I go on? Okay. The point is they have a very almost rigid outer cellular structure to them, including a cortex. We can see all the wonderful,
Mhm.
wonderful, beautiful structures to it. And you would think they would be better able to take the stresses. I want to say that one thing when I used to teach intro biology, I'm phasing into retirement, but um, I would often tell the students, all right, tell me how a phospholipid bilayer, that is to say the membrane around a cell, how does it deal with elevated
Right.
temperature?
Mhm.
What happens? How does it deal with lower temperature? And it's, for me, something where there isn't an outside structure to help protect that plasma membrane would seem to be more sensitive to temperature extremes, but that's not what's seen.
Yeah, I totally agree. And I'll say these amoeba can insist, meaning that they
Okay.
can like go into a protective state
Hmm.
at really high temperatures. So we see that when they're sort of above their replication limit, but surprisingly, they're not insisted um, at temperatures that are higher than these fungi that currently make up, our known thresholds for eukaryotes. So I have
What
no idea
temperature?
how they're doing it. We have no idea. That's where we're going, right?
But isn't that exciting?
It's super exciting. Yes,
Yeah,
it's super exciting.
students always say, well, why are you so excited? I get this look on my face when no one knows the answer. And I tell them that's what's exciting. If you knew
Yes.
the answer to everything, I suppose that would be okay. No hatred. I'm not a hater. Um, I don't drink haterade. You know the whole routine. But what I'm trying to say is that I want to know what happens next. I want to know
Absolutely.
more.
Yes. Okay. So let me give you a little kind of background on how we got them into culture in our lab and sort of what we've done to characterize them. And then I think you're right. All of the most interesting questions we have not figured out yet, which is exciting. And we're really curious. So we'll get to that. Okay, so wrapping up, we did that review paper. We saw amoeba were seemingly really good. We didn't know why, but we were like, well, now we need to go study amoeba and we need to go find them from the field because no one has cultures of them anymore. Right? So Beryl actually reached out to Gordon Wolfe, who is now emeritus, but was a professor at Cal State Chico for many years.
Yeah.
And he and his lab sampled at Lassen Volcanic National Park and did some of the early characterization of this amoeba, Tetramytis thermacidophilus, which is this cool amoeba that can withstand high temperature and low pH. So we're like, okay, well, this is a good target because it could use a lot more characterization, and we want to get a genome for it if possible. So we reached out to him. We ended up getting connected with Ken Stedman's group at Portland State University. They study viruses. I'm actually from Oregon too, so I was like, oh,
Oh,
perfect.
okay. Yeah.
um, so we invited ourselves along with Ken's group on a sampling trip to Lassen Volcanic National Park. And we sampled a bunch of really cool sites. So here's an image of Boiling Springs Lake. It gets to about 55 degrees Celsius. Its pH is 2.5, so really acidic. We sampled this other cool sort of set of geothermal features called Devil's Kitchen. which gets about 60 degrees Celsius, and the pH is 1 to 2.5. And then we also sampled this other area, which is this tiny little creek that's actually really boring for most biologists because it's on the way to Boiling Springs Lake, which is this huge, really, super acidic, bubbling lake. That's kind of the main show. And this little creek is on the way, and it's a pH neutral creek. So that's why we were interested in sampling it, because it's a really stark contrast from some of the really acidic sites. And this is the creek that we were able to culture this really high temperature amoeba from. So that's the picture there. And again, this work has really been spearheaded by Beryl Rappaport in my lab. Okay, so we collected all these samples. We had no idea if we were actually going to get anything. We brought them back to our lab. We followed some of Gordon's original designs for how to enrich the samples so that we could basically feed the amoeba. And it's really simple. You just put a wheat berry in a flask with a water sample from the site, right? That's it. So that's, as you can see from this image of our sort of approach to culturing, you can see a bunch of flask lines up. So those are different flasks that we sampled from Laston. And we checked them after a day. There was nothing. We checked them after 2 days. There was nothing.
Yeah.
We were starting to get nervous. After a week, we saw amoeba, and it was amazing. We were like, we didn't do this whole crazy field trip for nothing, right? And so that's what the cultures, when we clean them up and many transfers later, basically you get really, really dense amoeba in these flasks. And they're growing with bacteria that are feeding on the wheat berry. But the bacteria are basically a co-culture from the actual environmental site.
So that's generally called axenic growth. And it's one of the things that's always challenging. When I was learning microbiology, the idea of having to co-culture things was verboten. But it turns out even with bacteria, sometimes one type of bacterium will not grow except if another is present as well.
Yes.
And I think there's a lesson for all of us in life there.
That's right. Yes, you're totally right. So right now we have this high temp amoeba growing with a 12-member bacterial consortia. And we don't know if it needs the whole consortia, but it's happy, right? And that's perhaps why it hadn't been previously isolated, is because maybe people were trying to grow samples on just like E. coli or one type of bacteria.
So I have a nerdy question for you, may I?
Yes.
Does this thing survive cryopreservation?
Yes.
Did you cryopreserve a sample before you're passaging it?
Okay. So this is the
Bad
thing
question.
no, this is the thing that we want to do. So we went out in 2023. That was our first year. And then it took a while to kind of establish. We barrel cryopreserved it with our collaborators at LBNL, like fall 2024. But we went out in 2024. We were able to recover the same lineage from the field again, which gave us increased confidence. So we really want to do that in the future, because we know that passaging it is adapting it to our lab's high temp conditions.
And that's really important. I mean, I have to look you right in the eye. This is the camera, but I need to look you in the eye when I say this. The fellow who taught me about Dellavibrio, the predator bacterium, Sid Rettenberg, he's one of the people that isolated the strain that he worked with. He passaged it twice a week for over 20 years before any of it got frozen.
Wow.
So is it the same as the first thing? Probably not. But we didn't have the original, right?
Yeah, absolutely. And that's kind of one of the things that we think is a really cool science question, right? Like, how is it shifting under this different temperature regime? And can we actually do some thermal temperature experiments to
different parts of the creek, different parts of the ponds? Absolutely. Yeah. Same thing.
Yes. Yeah, totally agree. Yeah. So we have them in culture. We're really excited. And I have an image of two trees that just kind of highlight some of the diversity, because I keep talking about this high temperature one. But we actually have recovered 12 to 15 distinct lineages of amoeba, which is really exciting. It's like too much. We don't know what to do with all of them, right? And so they're mostly within two kind of major supergroups that are really distantly related. Heterolobosea is one, and you can see the gray highlighted ones are some of the lineages that we were able to get, including Gordon's Tetramitis, Thermocytophilus. So we're really excited about that. And then some within Amoebazoa. And the one I wanted to really highlight here are these DBS 1 and 2 samples. And we started growing them at an incubator at 57 degrees. We're like, gosh, they're really happy. Let's crank it up to 58. We kept growing them at 58. Let's crank it up, right? And then we hit 62. And we were like, phew. So I should say that the known, like experimentally observed temperature limit for any eukaryote for replication is 60 degrees.
Right.
And that has been observed in fungus and in a red algae. The theoretical limit for eukaryotes that was first proposed by Thomas Brock is 62 degrees. So we have found this amoeba that can actually replicate at 62 degrees, which is
That's amazing.
It's
So, you know what I wonder about, and I'm going to say something and you go, well, of course we looked. But when you look at some of the bacteria and archaea that live up to their limits, 80% of their protein are chaperones, because and so they're not really happy. They're kind of like, I'm growing a little bit. And I don't know if it's the same thing's true for your amoebae.
It's a great question. So I can say that 62 is definitely not their temperature optima. We're in the process of doing many, many growth experiments in a highly quantitative way in our lab and with our collaborators at LBNL to kind of prove independent cross-lab replication. And we're thinking that the optimum is around 54. So that makes sense. But they definitely double, we think, in about 2-ish days at 62. So they're fine.
I wanted to say something important. Several times you've mentioned, working with large research groups. You're talking about the Lawrence Livermore group, but also, and this is important for listeners and viewers to understand how collaborative science is, not simply even outside your group. I want everyone to notice the amount of ownership that's shared within your research group.
Absolutely.
And to you, it's like breathing. It's second nature. But I think some people have a funny idea, and I used to make a joke about it: one ego per lab, and it's not yours, right? And I'm delighted to say that that's no longer as true as it used to be. And it always delights me to hear visitors like you to the podcast talk about the wonderful work the folks with, and I'm not going to say they work for you, they work with you. They're collaborators with you.
And it's true, right? Like, this is true for this project, but also for other projects in our lab. My own research enthusiasm and directions are totally shaped by who's in the lab and what they're excited about.
I couldn't agree more.
Yeah. So it's a super, super fun project. And I'll say right now and I guess, one more thought on kind of our pipeline approach. And this actually brings me back to something that you were talking about earlier in terms of azenic cultures and how they can be really hard to get, right? And I think that's maybe part of the insight that we had as a group, which is because we've gotten so far with genomics and long-read sequencing and genome-resolved metagenomics and even eukaryotic genome resolved metagenomics, which is assembling eukaryotic genomes from complex environmental samples, right? We've made so much progress on that in the past 5 years. So I think our insight was kind of, and the challenge is actually getting enough euc DNA from the sample to be able to do that. Because typically in any type of environmental sample, you have way more bacteria than eukaryotes
Yeah.
because they're the top predators, right? Um, and so I think our insight was, hey, if we can enrich to get
Yes.
pretty dense amoeba, then we don't need to have perfect cultures because now we have all these other tools. So now we can use long read sequencing, which is what we did, and get pretty decent genomes. So we now are in the process of refining genomes for some of these organisms, but
Oh,
we're super, super excited by what we see so far, which is above 80% complete for many of them, which to bacteria people sounds pretty mediocre, but to euc people is kind of amazing.
I, I, no, I followed this for many years because I like single cell. I like protists.
Yeah.
And I've watched people. It's like blood from a stone getting stuff out. And in fact, this is one of the reasons I came up with this rule that some research topics appear to actively resist investigation. And that's probably how it feels some days, right?
Yes, absolutely. Totally. I like that. Um, yeah, so I think I don't think it's that we're doing anything particularly novel, right? We're just pairing methods that have come a long way
Certainly,
with something that's been done and is well tested for a long time.
certainly.
Yeah, and that's really exciting. And we've even detected giant viruses that we think are in these amoeba and lots of
Of
other
course.
potential, bacterial endosymbionts. So I've been talking a lot about amoeba, and I haven't shown you any pictures or videos yet. So I think it's probably time to do that. Um, and speaking of large collaborations, these images were taken with other collaborators at UCSF. Um, Dyke Mullen's lab group at UCSF has this really, they study amoeba, but also archaea, and they're really into actin. Um, and they have this really amazing microscopy setup where they have uh, a heated aperture or heated stage on the microscope. It's like a little chamber. It's from Bioptex, but basically it allows for micro control of temperature. So one of the huge challenges with this project is we want to get beautiful images of it, right? And we want to show it under the scope. But how do we do that? Because when we put it at room temperature, it insists. It actually doesn't move. It's unhappy. So anyways, we teamed, yes, it's like this. um, so we teamed up with Dyke Mullen's group at UCSF, and they were kind enough to let Beryl just take thousands of hours of, at least hundreds of hours of microbial video. So what you can see here is this high temperature worm amoeba. It's just moving around. Beryl has figured out that there's likely two motility types. So there's this pseudopodia, which is kind of like the classic blubbing, where it's worm-like, and then lamellipodia, which I kind of describe as looking like a little octopus. It's going like this.
Interpretive
I think
dance
we
is
need a new
necessary.
metaphor.
Mm,
It's the short. But basically, we were able to set the microscope heat to be highly controlled at 64
right.
degrees. And we can see that motility, they're active and feeding at 64 for hours. And when we keep them
Well,
in our
that
incubator, they don't insist at 64. They're not
good.
replicating, but they're active. So that's
But
kind of like this
they're
other,
still
yeah,
they're still hungry, is what I'm saying.
yes, exactly. And then we bring them back down, and then they start replicating again. Yeah, it's really, really wild. So we're like, how are they? And I think this gets back to your kind of key question, right? Like, how are they doing this? They're not using encystment. That comes at higher temperatures. When we put them at 70, that's what happens, right? But at 64, they're perfectly motile and happy and feeding. They're just not replicating. So, and then when they're at 66, Beryl has some amazing stills of these cells at 66 that start to look a little funky. So that we think is the intermediate transition to encystment.
Have you ever seen the video with mycoplasmas where you pop their you pop their cell membranes, you dissolve the DNA that they secrete to glue themselves down, and they have a motility system that looks like myosin.
Okay.
Pop, 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 pop. And then when you add a solution of ATP, the dead cells start walking around.
What?
Yes, I'll send you a link, and I'll put a link in the show notes. Now, the reason I bring this up is the structure of this looks very much the way that myosin does, where it has the two different sets as it cleaves ATP. And it's a machine. And that machine is very, very, very resistant. Eventually it breaks. And so any type of motility for an extremophile makes me think about that. Are they kind of leveraging themselves by having kind of a mechanical side to them, for lack of a better term? I don't know the answer, but you'll see if it's the case.
Yeah, that's super interesting. That's really interesting.
Because I don't think it has to do with reshaping the cytoskeleton. I really don't, because that's too flexible, especially when you look at how pseudopods work. Okay, that's my opinion. Okay.
No, all good things. It's such a question.
And we don't think about these things,
Yeah.
you know. I mean, I make jokes about eukaryocentrism and oxycentrism and collicentrism. Let's just say, for lack of a better term, mesophilocentrism, that we're only interested in about 35 to 37 degrees.
Right. Yeah. Yes, that's true. Yeah. I do think that, and this is just, I feel like I've been pulled into this new field, which is amazing. And also I'm just learning so much, right? So I think that and our lab has a lot to learn from people who study archaea at high temperatures and their adaptations, right? And I think that that's kind of some of the next directions that we're going is looking at if there are conserved mechanisms or similar mechanisms to how our archaea.
So interesting. I cannot express to you. In fact, so much so that I worry I'm stepping on things you want to say. So tell us more about what you want to do instead of listening to me go on and on.
Oh, no. So I think that, our first goal sort of from this effort is to basically feature this one amoeba that can replicate at 62 and is motile and feeding at 64, because that's pretty amazing. And and we have a genome and we have transcriptome data. The really hard part is, right, like even in the larger group of organisms that contains this clade, there's only one other genome. So it's like, well, how do we really make sense in light of other organisms? So we certainly have a lot of work to do there on the sort of comparative genomic side. We have some ideas about culturing and sequencing closely related mesophilic genomes as a next kind of place to go. But we're starting there, and then we're also working on characterizing in more detail these other fascinating organisms that we've been able to culture, many of which are thermoacidophilic. So that presents a whole new sort of area of questioning. And then we accidentally cultured a fungi. So Beryl thought it was a vampirellid, which are these weird, they're called vampire amoeba. If you want to talk about predator-prey dynamics in the microbial world, right?
Why are you not hiring artists to do these drawings?
Oh my gosh, well, Beryl does some amazing drawings, so I think we just need to, I need to let her have more time for these things, right? But I'm so excited.
I understand.
Well, what's the genome data say?
So Vampiro and you...
Vampirellids is what we thought they were, but they're not actually vampirellids. When we got this sequencing data back, multiple lines of sort of evidence from genomes, transcriptomes, all suggest that they're most closely related to chytrids. And I'm like, we're not a fungal group. We don't know what to do with this.
I hate those. So some chytrids are really nasty and are responsible for killing lots and lots of frogs, which I like.
Yes, I know.
And then there are bacteria that are on the skin of the frog that protects them from chytrids, and that's
Mhm.
just the coolest thing in the world.
So cool. So cool. Yeah.
And again, this is something that's interesting because you didn't expect it and it came your way anyway. Is it important? The number of times people have said it's not important, it turns out to yield like a lot of money, for lack of a better
Mhm.
term. It happens a lot. Who knows?
Yeah.
Right?
Yeah, and we have the sense, I say chytrids, but it's seemingly very distantly related to any
Okay.
known lineage. So we have an inkling that it might be important in understanding some deeper relationships, at least in the fungal tree.
I also think that, and I have strong feelings on this, so I'll keep those to a minimum. But some people want to say things like, does this work with high temperature amoebae have anything to do with issues involving human health? And I'm watching my expression, so it's not showing because I that shouldn't matter, but people always want to hear. Do you have thoughts on that?
Yeah, I mean, yes, absolutely. I think that's
It'd be funny if you said yes and nothing else. That would be hysterical.
just pause there. So a couple lines of thought here. One, amoeba, what we call free living amoeba, and you mentioned one earlier, Naglaria, are some of the most closely related heterolobosians to this free living amoeba. And there are lots of really fascinating hypotheses about how these amoeba might be sort of like Trojan horses for harboring
Yes.
bacterial pathogens like Legionella, right? The causative agent of Legionnaire's disease. So I think that understanding this group of uh, amoeba more comprehensively is really important to understanding sort of these potentially environmentally acquired infections and the movement and distribution of bacterial pathogens. So I think that's kind of one direct link to human health. Some of the closely related amoeba are actually also parasites, right? Like the brain-eating amoeba, um, and we understand very little about the mechanisms or why sometimes people are really harmed,
Yes.
right? And sometimes they're not. We have no idea why. But I suspect that building out the basic biology in this group of organisms can really shine some light on those types of questions as well.
I agree. And the business, any organism that, and I'm not gonna I don't want to be too general, but amoeba-like organisms like macrophages and neutrophils in us, which
Yeah.
are like tame amoebae, their whole raison d'etre is to eat things like bacteria.
Yes.
And then there are bacteria that learn to live inside them,
Right.
some of which cause diseases in us. So it should not surprise us that that is a series of ecological niches in which organisms could live. Just like with paramecium, there are different bacterial symbionts that live in different compartments.
Yes. Which is
Uh,
cool.
and so this would not be at all surprise. So this is like a Russian doll, perhaps,
Yeah. Yeah.
full of different things, just because of the size aspect of it, based on how big it is compared to bacteria.
Yeah.
And again, as you say, learning about that may have issues for human health. I am deathly afraid I'm going to get asked about hot tubs. That's my big fear here.
I have some thoughts on hot tubs.
Yeah.
I don't, and when I go into hot springs, which I still do occasionally, I don't put my head under
Yes.
and nose clips.
I don't blame you.
I'm not a medical professional.
No.
But
Nor do
those
you play one on TV. Right. That's exactly.
I do go into hot tubs and don't do those things, but I do think about it. It's hard not to
Yeah.
think about it. Yeah,
I,
and
I
then I
think
guess.
in
Oh, go ahead.
I think in a human hot tub, I think we have far more to worry about from bacterial contamination, to be honest with
Agreed.
you.
Agreed. Yeah,
Yeah.
totally agree. Yeah, and then I also think to may perhaps state the obvious for many people, but the potential discovery of novel enzymes and proteins and metabolisms that
Oh
could
yeah.
arise from these systems is quite high too. So I think there's a lot of
And
biotech
people
side work
people
there.
don't think about that. The fact that if you, and it's the same idea with any thermostable polymerase,
Right.
you can also find lipases that like low temperatures, which is why we don't have to boil our laundry to clean them anymore. So, yeah, I mean, who knows what you're going to find? And it's been there waiting for you and your crew.
Sometimes that's how it feels. um, yeah.
So, if you were to tell everybody what to remember about your work for today, what would you tell them?
Mm, ooh, great.
Besides the fact that amoeba are really awesome and metal.
That's right. I think it has to do something with uh, not overlooking the overlooked, right? I think
Yeah.
we can fully appreciate that most of the microbial diversity is bacterial and then archaeal, right? And nukes are just this tiny little lineage. uh,
Then the virologists get after me.
I know, it's true. It's
I
true.
can't win. Yeah.
I know, it's true. And I think that by studying these lineages across the eukaryotic tree, we can learn about fundamental eukaryotic cell biology, which is
Yeah.
really interesting.
I would be very surprised to learn that the first eukaryotes, the first organisms with a membrane bound nucleus, which is the definition I use,
Yes.
um, I would be very surprised that they weren't thermophilic based on what I know about how life evolved.
Yes. Well, we try to
So,
make that pitch, at least in grant applications.
sure. And
But I think it's
astrobiology as
real
well.
too, right? Yeah. Yeah.
Yeah. And so that whole idea is a good one. We see echoes of the past
Mhm.
in the gene sequences
Yeah.
that we find.
Yeah.
And I think that's what makes the work that we do so interesting and exciting.
Yeah. I totally agree.
So you did want to say something about your path to become a microbial scientist. And I can't say a microbiologist. Microbial scientist sounds like you're tiny. That's not what I mean, of course. What I mean is a person who studies microbial systems. So your path was an unusual one and you wanted to share it?
Yeah. So I will start off by saying that neither of my parents went to college. Um, and I'm extremely grateful to both of them for
Yeah.
nevertheless realizing the significant opportunity and sort of life-changing experiences that would present. They were always so pro education, and I'm
Yeah.
very grateful for that. And it was challenging figuring out college once I got there, right? And I know that you can probably say similar things. Um, and so, although I grew up loving science, I was quickly turned off by just kind of the sheer memorization that sometimes
Hmm.
courses present,
Yep.
and was actually a philosophy major, and I actually did double major in philosophy. And it wasn't until my junior year of college when I took intro bio course taught by Dr. Laura Katz at Smith College, who is a
I heard.
champion of protists. I should say microbial eukaryotes because she doesn't even like the term protist. um, and she just said at one of the first lectures in the course, "Hey, everyone, research, like do research on campus. If you want to get experience of what biology actually means,
Hmm.
do research. There are many labs that you can do research in. Email professors, go talk to them, right?" And I had no idea.
Yeah.
I had no idea. I had like maybe a vague awareness that this is a job that some people had on campus at that by that point, but I had like literally no idea that it was actually accessible to me.
Why would you have known?
Yeah,
Right.
right.
I mean, that's the thing. My I had the same background you're talking about pretty much. And I have an older brother. He went to college, but he wanted to be an attorney, which he should have never been. He should have been a historian, since he loved that.
Mm.
And at least the idea of being a lawyer made sense to my parents, right? But for me, and I'd always loved science and did science fairs and won awards. And I was just like you see now, which is often called too markish, but that's me.
Love it.
But I yield to no one in my enthusiasm. They had no idea. And I salute them every day, because they easily could have said, this is stupid and we're not going to put a cent into it.
Right. Yes, absolutely.
Right.
Yeah.
And if nothing else, listeners and viewers need to understand there are all kinds of paths to becoming a scientist. And even if you're not a professional scientist, perhaps you've learned about thinking in a scientific fashion from the classes that you
all
have.
All types of scientists.
That's right. I mean, one of the very best research students I ever had was completely uninterested in a career in research. She's a physician in Southern California, but her brain was exactly perfect for it, but that's not what she wanted to do.
Yep.
And what's wrong with that? That's her choice.
Yeah. Yep.
Yeah. So this has been like a wonderful episode. I love protists. I'm afraid to use, and don't repeat me to Dr. Katz. Lynn Margulis had a name for how you studied the field of protistal biology, and I can't pronounce it. It has like a C and a T next to it, but you know the word I'm looking for.
Protistology?
Sort of like that, yeah.
Or
That was very
protozoological.
She was very opinionated on that subject. Much missed. I only spoke to her a couple of times, but she petrified me every time, right?
Yes, absolutely.
But it's such an enormously wonderful world. And I'll put some links to some basic protistal biology that people can enjoy. The images, the videos are so wonderful. Your work has been wonderful to share. And I also know that I'm going to lasso you into another visit to talk about other research in your lab.
Perfect. Well, one, thank you so much. Two, you've given me some awesome science ideas. And I think having conversations is so fun to take a step back and then to think about new things to go look at. So I enjoyed it greatly.
So a secret that I think you've tagged me on is I do this podcast as much for me as for anyone else, because I get to find out things that I think are really cool every single episode. And today is no different.
Yeah.
So I want to wish you and your lab group the very best. I mean, you tell Beryl she needs to get some Jane Hartman glass like I have here.
I will tell her.
And I will buy some of Beryl's stuff.
Okay. Heard it here first.
I will. Okay. I can't wear the earrings, I got to admit.
Okay. Yeah.
I don't have the lobes for it. So there you go.
I hear that.
In any event, best wishes to you and your crew. Thank you so much for spending time with us today.
Likewise. Thank you, Mark. Such a pleasure.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with monumentally epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Angela Oliverio is assistant professor in the biology department at Syracuse University in Syracuse, New York. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.